Good morning, friends. Today, let us discuss about the hematology. I myself, Dr. Chandrasekhar Bangarwar, Professor, SRM State Ayurvedic College, Bareilly, Uttar Pradesh, will share some information about the blood with all of you. It is also called as hematology. The syllabus for the first year students expect by Central Council of Indian Medicine in the hematology is like this: hemopoietic system, composition functions of blood and blood cells, hemopoiesis, stages and development of the RBCs, WBCs and platelets, composition functions of bone marrow, structure, types and functions of hemoglobin, mechanism of the blood clotting, anticoagulants, physiological basis of the blood groups, plasma proteins, introduction to anemia and jaundice. For better understanding, let us revise these points in another manner so that you will able to understand it more easily. We will study composition functions of blood and blood cells along with plasma protein, then structure, types and functions of hemoglobin, then hemopoietic system, hemopoiesis, stages and development of the RBCs, WBCs and platelets, then composition and functions of bone marrow, mechanism of blood clotting and anticoagulants, physiological basis of the blood groups, introduction to the anemia and jaundice. So, let us start with the study of blood. Hemopoietic system or cardiovascular system. Because blood is one of the component of the cardiovascular system, the cardiovascular system consists of three interrelated components: blood, heart, and blood vessel, out of which today we are going to study about the blood. The branch of science concerned with the study of blood and blood forming tissue and disorders associated with the blood is called hematology. In simple language, hematology is study of blood about which we are going to study detail. In today's lecture, due to the limitation of the timing, which is hardly up to 30 minutes, we will able to see the blood physical characters, then composition, blood plasma, plasma proteins, red blood cells and white blood cells. So, what is blood? Blood is a connective tissue. This blood is composed of the two parts, plasma and cells. Basically, blood is red colored connective tissue running through the blood vessels in whole body in circulatory system. We should remember the physical characteristics of the blood. Number one, blood is a denser and more viscous than water and feels slightly sticky. It has red color due to the presence of hemoglobin, a red colored pigment in RBCs. Blood constitutes about 20% of the extracellular fluid in the form of plasma, and total blood occupies or constitutes 8% of the total body mass. Then number 3, the blood volume is averagely 5 to 6 liters in an average size adult male and 4 to 5 liters in an average size adult female. Number 4, the temperature of the blood is 38 degrees Celsius, 100.4 degrees Fahrenheit, about 1 degree Celsius higher than oral or rectal body temperature. And last, we should remember the blood has slightly alkaline pH ranging from 7.35 to 7.45. Composition of the blood. Blood consists of two parts, blood plasma and blood cells. Out of these, blood plasma makes up 55% of the blood and blood cells makes up 45% of the blood. There are three types of the cells in the blood: red blood cells or erythrocytes, the white blood cells or leukocytes, and platelets or thrombocyte. First of all, we will see something about the plasma. What is blood plasma? Plasma, a liquid portion of the blood, is matrix that contains dissolved substances and form elements which are mostly cells. This plasma is about 91.5% water and 8.5% of the solutes, most of which 7% by weight are proteins. Some of proteins in plasma are also found elsewhere in the body, but those which confine to the blood are called plasma proteins. If you see the composition of the blood plasma, you will see that water is 91.5% of the total blood plasma. Proteins are 7%. In the 7% of the proteins, 54% are albumins, then 38% are globulins, and 7% are fibrinogen. In addition, plasma contains some electrolytes, some nutrients, some regulatory substances such as enzymes and hormones, some gases, and waste products such as urea, uric acid, creatinine, ammonia, and bilirubin. Regarding water, is a liquid portion of the blood, act as a solvent and suspending medium for a component of the blood absorb, transport, and releases heat. Regarding plasma proteins, some of the proteins in plasma are also found elsewhere in the body, but those confined to the blood are called plasma proteins. Among other functions, these proteins play a role in maintaining proper blood osmotic pressure, which is an important factor in exchange of the fluid across capillary walls. Then hepatocytes synthesize most of the plasma proteins, which includes albumin, globulin, and fibrinogen. The protein has got many functions: exert colloid osmotic pressure, which help maintains water balance between blood and tissue and regulate blood volume. Albumins are the smallest and most numerous plasma proteins produced by liver, function as transport protein for several steroid hormones and for fatty acid. The globulins are produced by liver and plasma cells which develop from B lymphocyte. This lymphocyte is a type of WBC. Certain blood cells develop into cells that produce gamma globulin, an important type of globulin. These plasma proteins are also called antibodies or immunoglobulins because they are produced during certain immune responses. Antibodies help attack viruses and bacteria. Alpha and beta globulin transport ion, lipids, and fat soluble vitamins. Fibrinogen is produced by liver. Fibrinogen plays important role in blood clotting. In the blood plasma, other solutes are present in 1.5%, includes electrolytes like inorganic salt, positively charged ions include sodium, potassium, calcium, magnesium, negatively charged ions include chlorides, phosphate, and carbonates. These electrolytes help maintain osmotic pressure and play essential role in the function of the cell. In the blood plasma, nutrients are also present. Basically, they are the product of digestion passed into blood for distribution to all body cells. These includes amino acids of protein digestion, glucose of carbohydrate digestion, and fatty acid and glycerol of lipid or fat digestion and vitamins and minerals. Plasma contains gases, oxygen, carbon dioxide, nitrogen, whereas more oxygen is associated with hemoglobin inside RBC, more CO2 is dissolved in blood plasma. Nitrogen is also present but has no known functions in the body. Some regulatory substances like enzymes, which are produced by the body cells, catalyze chemical reactions. Hormones produced by the endocrine glands regulate metabolism, growth and development in the body. Vitamins are cofactors for enzymatic reactions. Some waste products are also present in the plasma. Most are the breakdown product of the protein metabolism and are carried by the blood to the organ for excretion. Includes urea, uric acid, creatine, creatinine, bilirubin and ammonia. Let us see the functions of the blood. Blood has got various functions in the body, but these functions can be categorized into three categories: transportation, regulation and protection. Regarding transportation, blood transport oxygen from the lungs to the cells of the body and carbon dioxide from body to the cells and to the lungs for exhalation. It carries nutrients from the gastrointestinal tract to body cells and hormones from endocrine glands to other body cells. Blood also transports heat and waste product to the lungs, kidneys and skin for elimination from the body. Regulation, circulating blood help maintain homeostasis in all body fluids. Blood help regulate pH through buffers. It also help adjust body temperature through the heat absorbing and coolant properties of the plasma and its variable rate of flow through the skin, whereas excess heat can be lost from the blood to the environment. Blood osmotic pressure also influences the water content of the cell, mainly through the interaction of dissolved ions and proteins. Third function is protection. Blood can clot, which protect against its excessive loss from the cardiovascular system after an injury. In addition, WBCs protect against disease by carrying on phagocytosis. Several types of the blood proteins, antibodies, such is and unique from person to another person, as are skin, bone, and hair. Now regarding blood cells, there are three types of the cells present in the blood: red blood cells or RBCs or erythrocytes, WBC or white blood cells or leukocytes, and platelets or thrombocytes. Let us see about red blood cells or erythrocyte. RBC contains hemoglobin, which is a pigment that gives its red color. Red blood cells or RBC contain the oxygen carrying protein, hemoglobin, which is a pigment which gives whole blood its red color. Regarding hemoglobin count, we should remember that normal hemoglobin ranges are infants 14 to 20 grams per 100 ml of the blood, adult females 12 to 15 grams per 100 ml of the blood, and adult males 13.5 to 18 grams per 100 ml of the blood. Regarding its structure, RBCs are biconcave discs with the diameter of 7.2 μm. RBC lack a nucleus and other organelles and can neither reproduce nor carry an extensive metabolic activities. The cytosol of the RBC contains hemoglobin molecules which are synthesized before loss of the nucleus during RBC production. Functions of RBCs. The main function of the RBC is to carry oxygen. This function is done by the hemoglobin present in the RBCs. RBC also carries small amount of carbon dioxide produced during the cellular metabolism or cellular respiration as waste product and carries it towards excretory organs for elimination. RBCs are highly specialized for their oxygen transport function. Because mature RBCs have no nucleus, all their internal space is available for oxygen transport. Moreover, RBC lack mitochondria, that means RBC do not have mitochondria in the cell, and generate ATP anaerobically, that means without the help of oxygen. ATP can be generated aerobically by using oxygen or anaerobically by not using oxygens. Here anaerobic ATP generation is done, so they do not use up any of the oxygen they transport. Each RBC contains 280 million hemoglobin molecules. A hemoglobin consists of four non-protein ring-like pigments called heme or iron and proteins called as globin, which are four polypeptide chains, 2 alpha and 2 beta. Oxygen picked up in the lungs is transported bound to the iron of the heme group. Hemoglobin releases oxygen which diffuses first into the interstitial fluid and then into the cell. As blood flow through the tissue capillaries, the iron-oxygen reaction reverses. Hemoglobin also transport about 13% of the total carbon dioxide, a waste product of the metabolism. As blood flows through the lungs, the carbon dioxide is released from the hemoglobin and then exhaled. In addition to its key role in transporting oxygen and carbon dioxide, hemoglobin also plays a role in regulation of the blood flow and blood pressure. The gaseous hormone nitric oxide produced by the endothelial cells of the blood vessels bind to the hemoglobin, which is then released. This nitric oxide causes vasodilatation and increase in the blood vessel diameter that occurs when smooth muscles in vessel wall relaxes. RBC count. To maintain normal RBC count, RBCs they are produced at the rate of 2 millions per second. Normal RBC count, a healthy adult male has about 5.4 millions of RBCs per microliter of the blood, and healthy female has 4.8 millions of RBCs per microliter of the blood. RBC life cycle. RBCs live only about 120 days because of the wear and tear their plasma membranes undergo as they squeeze through the blood capillaries. Without a nucleus and other organelles, RBC cannot synthesize new components to replace damaged ones. The ruptured RBCs are removed from the circulation and destroyed by the fixed phagocytic macrophages in the spleen and liver. RBC life cycle is maintained as follows. Macrophages in the spleen, liver and red bone marrow, phagocyte, ruptured and worn out RBC, that is number 1. Number 2, the globin and heme portion of the hemoglobin are split apart. Number 3, globin is broken down into amino acids which can be reused to synthesize other proteins. Number 4, iron removed from the heme portion is in the form of Fe3+, which associate with the plasma protein transferrin. In muscle fibers, liver cells and macrophages of the spleen and liver, Fe3 again detaches from the transferrin and attaches to an iron storage protein called ferritin. Number 6. Upon release from the storage site of for absorption from GI tract, Fe3 reattaches to transferrin. The Fe3 transferrin complex is then carried to the bone marrow where RBC precursor cells takes it up through the receptor mediated endocytosis for use in the hemoglobin molecule, whereas amino acids are needed for the globin portion. 8. Erythropoiesis in red bone marrow results in the production of RBCs which enter the circulation. Number 9. When iron is removed from the heme, the non-iron portion of the heme is converted to biliverdin, a green bile pigment, and then into bilirubin, a yellow-orange-like pigment. Bilirubin enters the blood and is transported to the liver. Number 11. Within the liver, bilirubin is secreted by the liver cells into bile, which passes into the small intestine and then into the large intestine. Number 12. In the large intestine, bacteria converts bilirubin into urobilinogen. Some urobilinogen is absorbed back into the blood, converted to a yellow pigment called urobilin, and excreted in the urine. Number 14 of the RBC life cycle, most urobilinogen is eliminated in the feces in the form of brown pigment called stercobilin, which gives feces its characteristic color. Now let us see about the WBCs. The difference between RBC and WBC, unlike RBCs, WBCs or leukocytes have nucleus and do not contain hemoglobin. Now these WBCs are classified as either granular or agranular depending on whether they contain conspicuous chemical filled cytoplasmic vesicles that are made visible by staining. Granular leukocytes includes neutrophils, eosinophils and basophils. Agranular leukocytes includes lymphocytes and monocytes. So, in this way, there are 5 types of the WBCs: neutrophils or polymorphs, eosinophils, lymphocytes, monocytes, and basophils. The percentage is neutrophils are 60 to 70%, eosinophils 2 to 6%, lymphocytes 20 to 45%, monocyte 2 to 8%, and basophils 0 to 1%. Let us see one by one type of WBC. Neutrophils. The granules of neutrophils are smaller, evenly distributed and pale headache in color. The nucleus has 2 to 5 lobes. Older neutrophils have many lobes in nucleus, so they are also called as polymorphs. Eosinophils. A large uniform sized granules of an eosinophil are eosinophilic. They stain red orange with acidic dyes. The granules usually do not cover or observe the nucleus, which most often has 2 to 3 lobes connected by either a thin strand or thick strand. Lymphocytes. In lymphocyte, the nucleus of the lymphocyte is round or slightly indented and stained dark. The cytoplasm stains sky blue and forms rim around the nucleus. Monocytes. Monocytes are 12 to 20 μm in diameter. The nucleus of monocyte is usually kidney shaped or horseshoe shaped, and the cytoplasm is blue-gray and has a foamy appearance. Now, blood transport monocytes from the blood to the tissue. They enlarge and differentiate into macrophages. That is the speciality of the monocyte. Some becomes fixed macrophages, which means they reside in a particular tissue. Examples are alveolar macrophages in the lungs, macrophages in the spleen, and stellate reticuloendothelial cells in liver. Other becomes wandering macrophages, which roam the tissues and gather at site of infection or inflammation. Basophils are the round variable size granules of the basophils are basophilic. They stain blue-purple with basic dyes. This is about the 5 types of the WBCs. Regarding lifespan of the WBC, in healthy body, some WBCs, especially lymphocytes, can live for several months or years, but most likely only a few days. During a period of infection, phagocytic WBCs may lie only a few hours. Functions of WBCs. The skin and mucous membrane of the body are continuously exposed to the microbes and their toxins. Some of these microbes can invade deeper tissues to cause the disease. Once pathogen enters the body, the general function of the WBC is to combat them by phagocytosis or immune responses. To accomplish this task, many WBCs leave the bloodstream and collect at the point of pathogen invasion or inflammation. Once granulocyte and monocyte leave the bloodstream, to fight injury or infection, they never return to it. Lymphocytes, on the other hand, continually recirculate from the blood to the interstitial space of the tissues to lymphatic fluid and back to the blood. Only 2% of the total lymphocyte population is circulating in the blood at any given time. The rest are lymphatic fluid and organs such as skin, lungs, lymph node, and spleen. Now, how these WBC react with the infection? Neutrophils and macrophages are active in phagocytosis. They can ingest bacteria and dispose the dead matter. Several different chemicals are released by the microbes and inflamed tissue attract phagocytes, a phenomenon called as chemotaxis. Among the WBCs, neutrophils respond more quickly to the tissue destruction by bacteria. After engulfing a pathogen during phagocytosis, a neutrophil unleashes several chemicals to destroy the pathogen. These chemicals include enzyme lysozyme, which destroy certain bacteria. Neutrophil also contains defensin, protein that exhibit a broad range of antibiotic activity against bacteria and fungi. Monocyte takes longer to reach site of infection than does the neutrophils, but they arrive in large number and destroy more microbes. Eosinophils leave the capillaries and enter tissue fluid. They are believed to release enzymes such as the histamine that combat the effect of histamine and other mediators of inflammation in allergic reactions. High eosinophilic count often indicate an allergic condition or parasitic infection. At the site of inflammation, basophils leave capillaries, enter tissues and liberate heparin, histamine and serotonin. These substances intensify the inflammatory reaction and are involved in hypersensitivity reactions. Like basophils, mast cells also liberate mediators in the inflammation, including heparin, histamine, and proteases. Now, lymphocytes are the major soldiers in the immune system battle. Three main types of lymphocytes are B cells, T cells, and natural killer cells. B cells are particularly effective in destroying the bacteria and inactivating their toxins. T cells attacks viruses, fungi, and transplanted cells, cancer cells, and some bacteria. Immune responses carried out by the B cell and T cell help combat infection and provide protection against some disease. T cells are also responsible for the transfusion reaction, allergies, and rejection of the transplanted organs. Natural killer cells attack a wide variety of the infectious microbes and certain spontaneously arising tumor cells. An increase in the number of circulating WBC usually indicate inflammation or infection. A physician may order a differential WBC count to detect an infection or inflammation. WBCs are far less numerous than RBCs, about 5,000 to 10,000 cells per cubic millimeter of the blood. RBCs therefore outnumber WBCs by about 700 to 1, that is its ratio. Leukocytosis, an increase in number of WBCs about 10,000, is normal protective response to stresses such as invading microbes, strenuous exercises, anesthesia, and surgery. An abnormally low level of the WBC is termed leukopenia. It is never beneficial and may be caused by the radiation, shock, and certain chemotherapeutic agents. This is about the WBCs. I will like to suggest all of you just to go through all the things in details, collect more information from other books. Any difficulty, you can call me or you can contact me. In the next sharing of the video, we'll start with the study of platelets, the hemostasis reaction, blood groups, and bone marrow. Thank you. Thank you very much.